I want to thank the opportunity to talk here. So I am going to try to address probably a lot of questions and and not too much answers about how to use to use quantum biology in some of the problems that we have been working in the last 20 years. I want to start with this is a book I read when I was maybe a teenager, and I was very impressed. In fact, I was not a teenager. I was in Champaign-Urbana. I read, and I bought it from my advisor there, Gregory Weber. And since I was working, just had finished the medical school, and I was trying to address protein thermodynamics, and sometimes you have to you need a new physics, but you needed to look in a different way. And I'm sure that probably James Watson read this book when he was a teenager. He was 23 years old when he joined Francis Crick and published this model of DNA, and based on some structured data that revolutionized and all the story. And in the last years, there has been a kind of we have for many years worked with protein folding, protein interacting, protein interactions, and protein aggregating. But in the last 10 years, a lot of focus of different science has been on this, how eventually proteins get together in making these membraneless organelles, and that is very important for function, especially in the nucleus. You need it, for example, in the nucleolus, you need it to assemble ribosomes. You need a lot of interacting, putting together these proteins and so forth. So this is quite a new paradigm. And of course, we know that the formation of these phase-separated liquids, just like separating oil from water, and in fact, it's a protein separating, and this is quite important. And eventually, and this is important for cell function, and but eventually this goes wrong, and you have the aggregate phase transition. You have aggregates, and you have amyloids causing Alzheimer's, Parkinson, etc., and also cancer that I'll be talking a little bit while. And when you try to understand how the mechanism, how these proteins get together and stay together for a while, that is very dynamic when you have these liquid droplets. It depends a lot on this different interactions. For example, amino acid residues like pi-pi interactions, cation pi interactions, usually interacting with nucleic acids, and sometimes non-specifically. That's probably how it's hard to address by different tools. And so of course, on the other side, I've been working a lot on using structural biology, especially NMR, but also other techniques. And so here just some advertisement of an NMR center that we have at the Federal University. We have several equipments doing liquid and solid state NMR, etc. And NMR is very important because you can not only get structure, but you can do dynamics. Of course, has some problem. For example, cannot work with proteins very big. Well, work with cells not that easy. You can have some cells, and but again, you have the problem of concentration. On the other side, I want to stress, Mauricio did a very good job, and so I don't need to go through all these, but it's for biophysicists, very important to use fluorescent techniques. That's in fact how I got my most of my training in Champaign-Urbana with Gregory Weber, and a colleague here, also Sergio, got that many years ago, a few years ago, Sergio. So anyway, so it's very important. So as I mentioned, we for many years we have tried to understand some of these, how protein folding eventually goes wrong and make these aggregates. We now know that before making the aggregates, probably they goes into these liquid droplets. And so I'll give some examples. And another dimension that we have used along all these years is to use high pressure. I mean, because with biophysics, you can use some chemical perturbation, can use the temperature. And we learned that pressure is very important because it deals a lot of with you can manage especially the hydrating or dehydration, and it's a very subtle way to perturb the equilibrium and get a very good thermodynamic information. And of course, because especially with the aggregates, one can observe, for example, changes in hydration. And these are all probably quantum effects because sorry, we'll go a little bit back. Can I go back? Sorry. Oh, okay. Okay, got it. You have proteins dissociate or denature by pressure because you have cavities. You cannot satisfy all the van der Waals interactions, and also have hydration, and this is like a dipole induced interactions, and you have all these interactions, and that's why pressure is quite a good way to look through all these reactions. We have used them in many cases. And especially for protein fold, it's quite interesting because we can populate these different species and look at them by NMR, by fluorescence, etc. And we won't go through too much of this detail, but just show that we can do, for example, this is data from alpha-synuclein. If we have an amyloid fiber, we can dissociate the fiber with pressure and get information by high pressure NMR and get both structure and dynamic data. And we can go through all these and see that you have here the aggregates, and you dissolve the aggregates with pressure. So, but then I want to go to my proof of work, that is p53. That is a protein that is very important as a tumor suppressor, but is mutated in more than 50% of the different cancers. And if of course you have a mutation, p53 doesn't work, and then you have the lack of a function. But the problem, since is that somatic mutation, one can question, well, why the other one doesn't do the work? And so we start to think about that. So we decided to work with this protein, especially with the DNA binding domain, because it's it has a single tryptophan, so you can probe the changes in the naturation. And we found that many years ago, about 20 years ago, we found that it aggregates as it's get denatured, it aggregates, and the mutants get more. Have more. You need lower temperatures and lower pressure to get it denatured. So then we proposed that maybe aggregation similar to what we have in some amyloid fibers would be behind not only the lack of a function, but also to eventually to some gain of a function, because you could have the mutant p53 inducing the wild type p53 get into that aggregates. So we decided, well, but that was using high pressure initially, but also we could observe using 37 degree. If we wait long enough, we have the wild type 3 are getting aggregates and getting this kind of amyloid fibers. And if you have the mutant, you need much, it's much faster and at a higher proportion. And then we decide to test, well, if Sisson were proposing that maybe the main problem when you have a mutation of PIF3 is that it would act as a seed and eventually convert the wild type into these fibrils. So we decided, well, to test this in vitro still, and we could find that indeed wild type takes a long time to wait. That's of course is in vitro. Remember that in cells, the lifetime of a PIF3 is only 4 minutes. So, but if you have a seed that is accumulating, then it will accelerate these aggregation process, and you have the aggregation. Then the next time, of course, was, well, okay, you have that in vitro. That's very beautiful. But how about the cell? So we use these probes that can identify amyloid aggregates inside the cell. And then we found that one can see that if it's not aggregated, you don't have the signal. So that's breast cancer biopsies. One can see that when you have the mutation, that's the labeling of a PIF3, and that's the aggregate PIF3. So you have this co-localization. And in another article, we looked about 80 different biopsies and a good correlation between a lot of aggregate PIF3 with correlating with the mutant PIF3 inside the cell. So this was with breast cancer cells, then went a little bit further with glioblastoma cells. That's the same kind of data showing this red here, these aggregates. And in this case, we tried to connect first the correlation that we had seen before, that when you have the mutation, you have more aggregates. And when you use pressure to try the stability, one can see that this mutant M237I is much less stable and it aggregates much faster, as one can see in this curve, this showing this aggregation. And here this particular glioblastoma was kind of interest because it has some gain of function. It induced a resistance to a chemo resistance to treatment. And then one of the questions, well, okay, how about living cells? And we use fluctuation microscopy to localize inside the cells without using antibodies. We could see the formation of these oligomers in the mutant cells and not in the wild type cells. So we proposed many years ago, we and other, and people start to find in different type of cancers the formation of these aggregates. Not only that, but that could have kind of a prion-like effect, eventually not only converting inside the cells, aggregating with other transcription factors, but also transmitting from cell to cell, like it occurs also in some in Alzheimer's and in some other neurodegenerative disease. So we decided to ask one more question. Okay, we could see that there was an aggregate here, but how it gets into the aggregate. So we start to see the literature and a lot of papers showing the formation of phase separating proteins, and we found that in fact the PIF3 can also make these liquid droplets, and especially when you have the mutant, we have the formation of aggregates or phase transition, and we could see that both in cells and in vitro. So we use this different mutants. We use the glioblastoma mutant, and we also use an hepatocarcinoma mutant. And one can see when you have the droplets, if you do a bleaching, you do a photo bleaching, and you see the recover, one can see that the wild type recovers much more than the wild type, indicating that you have more rigidity, so you don't have diffusion. And of course, what was important that was to look that in the cells, particularly in the nucleus, and when you have the mutant, it's particularly concentrated in the nucleolus, and you can see that you have much less recovery of the photobleaching in comparison to the wild type. And so our lab is trying to understand both the level at the molecular level, but also the cellular level, how these structures are. And eventually we know that you have this kind of a gain of function in some, see, 5 minutes. In some, of course, in neurodegenerative disease, we have the gain of function means killing the cell. But here in tumor cells means that making the cells to live forever and to have to be more metastatic, etc. So we are trying. We're trying to do the cryo-EM of these aggregates, and that's work that we're still trying to do, but I think it would be very important to understand how these aggregates that coming out of these kind of droplets are interacting probably with other proteins and with other RNAs. So that's a version of that's one review that we propose that in fact is PIF3 has a very chameleonic, it's like a chameleon. And of course, at the functional level, it would act probably in these droplets. In fact, there's a paper that just appeared last week by the group from Denmark, and showing that probably most of the function of PIF3 depends on going to these droplets. So that would be like a Dr. Jekyll good way of having PIF3 being so chameleonic, but that pays a price. If you have a mutation, it can become this Mr. Hyder interacting, including with P73, other tumor suppressors, and making the protein quite bad. So going to the last few minutes, just showing that we can, our main idea is to try to use aggregation of PIF3 as a target to use some compounds to prevent the aggregation, but also to rescue the function of the mutant. We are trying different compounds. Resveratrol is very good using wine. Unfortunately, well, we could see an effect, but using 100 micromolar, that means that we would need to drink 10 liters of wine. That's probably not good, but one can see that is very well. The idea is to find some derivatives. We have also looked for some mycoreceptors compound. This is one compound that is now in phase 3. People didn't know exactly what's the mechanism, so we show that most of the effect is indeed inhibiting aggregation. One can see here red oligomers, and then as you increase primer 1, it goes, so it disappears, and also we don't see it more. So we propose this mechanism. We have another compound in the lab that we are filing the patent. Is things in Brazil is not that easy, but we're filing the patent because we want to publish the paper. And this compound here, RCTP17, has a very good inhibition action. For example, in comparison with Prima 1, we use about 5-fold less concentration. So, and then the compound, one can see that in xenograft animals, it reduces a lot the size of the tumor and prevents the growth of the tumors. And in fact, many groups, of course, I don't have time to show the data of other groups, have developed compounds to target the PIF3 aggregation. And recently we had this kind of a perspective review in Nature that showed that indeed aggregation PIF3 could be one of the important targets. And pretty much my talk, I think, just want to stress, of course, we are very anxious to more, of course, we want to develop the cure of cancer of course, you don't develop acute cancer. Cancer is a very complex disease, but we want to understand that at the molecular atomic level, these interactions, and we do believe that those depend a lot on quantum mechanical interaction, especially among the aromatic amino acids used for example, for TDP-43 involved in amyotrophic lateral sclerosis. It depends a lot on the tryptophans. So with that, I want to thank all the group, a lot of people, and thank you.